So it is my great pleasure to introduce Fei Chen. He is a core institute member at the Broad Institute of MIT and Harvard and assistant professor in the Department of Stem Cell and Regenerative Biology at Harvard University. During his PhD at MIT, he co-invented expansion microscopy, a technology that allows for super resolution imaging of biological samples with conventional light microscopes. And then as an independent fellow at the Broad, he led a group which combined microscopy with next generation sequencing through in situ sequencing and Slide-seq technologies. And then he continues to do the same with his new group at the Broad with novel molecular and microscopy tools. So he has received several awards, including the NIH Director's Early Independence Award and the Allen's Distinguished Investigator Award. And we are very happy to have him as one of our keynotes today. So welcome, Fei.
Thank you. Good afternoon. I want to thank the organizers for putting together a great virtual conference. And I'm really excited to be here today to talk to you about Slide-seq, a technology we've developed for spatial genomics. And I want to start off with this video. This is every cell in a developing Drosophila embryo from shortly after its fertilization to when it's practically walking off the microscope. And I think I've always been drawn to this video because it beautifully illustrates and shows off the wonder of our natural world, and it also showcases the immense spatial and temporal coordination that exists in biology. In the Drosophila, developmental process is stereotyped. We can go in time and time again to label different cells and molecules to build up an understanding of how this process works in time and space. But for many other biological systems, such as the mammalian brain or in disease, we don't have the luxury of such stereotype processes. And what we really need are tools that allow us to study the molecular mechanisms governing spatial temporal biology. Now, one tool that has recently been quite powerful for studying molecular mechanisms themselves, and I'm sure you guys have heard a lot about in this conference, is single-cell genomics. And single-cell genomics, and in particular single-cell transcriptomics, is powerful because it's comprehensive. And it gives you a look at the cell types and states within a tissue and within cells. And from that, you can get a sense of the molecular programs that are existing within these cells. However, fundamentally, I think we have to remember that to apply single-cell transcriptomics, we have to dissociate a tissue. And when we do that, we lose the spatial and temporal context about where the cells come from. So we take a piece of tissue and we put it into kind of, we grind it up, we put it into a tube, and we come out with this single-cell slurry, and then we put that into a sequencer. And while we get to measure individual cells, we lose the context of who their neighbors were, what their interactions are. And I think in biology, this sort of structure of the tissue is going to be very important for understanding tissue function because cells communicate with each other, they self-organize to form functional structures, and more importantly, I think cells and tissues are dynamic as well, right? They migrate, they differentiate, they divide, and we lose that context as well when we dissociate. And so today, really, I'm going to focus on talking to you about a tool that we've developed to address this loss of spatial context when performing single-cell sequencing. And this tool is called Slide-seq. And what it enables is genome-wide expression profiling in tissues at about 10 micron resolution. This is a joint effort by my lab and Evan McCoster's lab at the Broad. We're both at the Broad. And the big picture of Slide-seq is that it effectively lets you do untargeted single-cell sequencing. And at the same time, it lets you know where those cells came from in your tissue. Now, our motivation from building this technology came from two critical kind of areas that we really think is driving this sort of need. And one is, the recent revolution in single-cell profiling technologies has really enabled us to collect large atlases, cellular atlases, which let us identify cell types as a function of molecular profiles. However, we really need a way to perform high-resolution mapping of those cell types and those gene expression programs into tissue in stereotype cases, such as in organ systems, for example, here in the brain, but also in non-stereotype cases, such as in disease. And that leads us to the second motivation, which is I think we really need ways to relate gene expression to tissue pathology, because for a long time I think we've been able to diagnose diseases by looking at the pathology under a microscope. For example, here on the right, in cancer, pathologists can look at different patterns of cells which are chromogenically stained, just stained with a couple of different colors, labeling the structures, and you can start to identify patterns which predict the characteristics of disease. However, we started to understand the molecular mechanisms perhaps behind, for example, different types of tumors, and we really like to connect that with our pathological understanding or morphological understanding of pathology, and then we might be able to learn, when you have a certain pathological pattern, what are the underlying molecular mechanisms. The other is that, as we've explored single-cell biology, we've started to uncover cellular programs which are important in disease pathologies. For example, in this beautiful paper, there were Karen Shaw et al. Found disease-associated microglia, which in the brain, which kind of surround amyloid plaques. And we'd really like to know, for example, what are the other cells, and this is, just a fluorescent histology image showing a very specific marker, but we really like to know what are all the other cells surrounding these cells and what are the molecular programs underlying those cells. So that's a broad motivation. We want to be able to connect gene expression to pathological states within tissues. And so we sought out to develop technology with some characteristics that we thought would be important both for our ability to apply the technology, but for all those people to use it. And really, I think we want to leverage the ability to do unbiased genome-wide profiling, which is what I mean is that we want to be able to sample the transcriptome, the genes that are being expressed. And we want, as we've discovered, I think the single cell resolution aspect where you can uncover the heterogeneity of gene expression programs between different cell types is very important. And so I think this suggests that we want to capture gene expression at relatively high spatial resolution. About, a mammalian cell in tissue is about 10 microns. And I think we also, want the technology to be relatively easy to use, as in the data collection should be relatively straightforward. And so how does Slide-seq work? Well, in Slide-seq, we deposit individually barcoded 10 micron polystyrene beads onto a monolayer onto a surface. And I think the key here is that each bead has a clonal sequence barcode. And what do I mean by that? That means that, what it means is that each one of these beads has a spatial bead barcode. And if that barcode is a unique DNA sequence that's the same for each bead, but different among different beads. And what we do is we deposit these beads randomly onto the surface of an array. And this is a 40 millimeter cover slip. And each one of these white circles is about a 3 millimeter diameter array with about 100,000 beads. And once these beads are, and you can see here kind of a zoom in image of the beads sitting on the array. And so once these beads are deposited, we then take a microscope and sequence the clonal bead barcodes for each bead in the array. And I won't go into details exactly how we do that, but in effect, it allows us to read out the base identity for each bead, whether it's an A, C, G, or T, in these spatial bead barcodes. And that lets us generate an XY location that corresponds to each one of these unique clonal bead barcodes. And this process of making the arrays can be done up front. And so that allows us kind of scalably manufacture them before performing the experiment. And one of the key strengths of Slide-seq is that once the arrays are made, it's very easy to actually then perform the experiment. So we can take these arrays which we've stored, then you take fresh frozen tissue, and this is kind of like, you know, you can dissect out here, for example, a piece of mouse brain, and then you can section it on a cryostat. That's kind of like a fancy deli slicer. You can cut a very thin section of this frozen array on to the beads. And then in a very simple protocol, we can generate sequencing libraries. And this is very similar to the library preparation approach in single cell sequencing. And these sequencing libraries, what happens is that the tissue, the RNA from the tissue is captured onto the beads, right? And then they're effectively barcoded by the XY locations of the clonal bead barcodes, and then we PCR it up. And when you sequence these libraries, you get data very much like single cell libraries. For each bead barcode, you have this matrix for a vector of bead barcodes, I guess. You have this matrix where along each row is a unique bead barcode, and each column is the kind of the counts of the genes across the transcriptome. But now if you remember, we have the spatial location of each bead barcode. And so then we can just match them up. And now you have a spatial location for each of these bead barcodes, and thus each read in your gene expression matrix. Now, what does the data look like? Actually, Slide-seq data has some of the same structure as single cell data. And so we can apply similar computational tools, such as dimensionality, reduction, and clustering. And so here we ran Slide-seq on the mouse hippocampus. And on the very left here, we have clustered the Slide-seq beads and annotated the clusters with cell type identities. And so here we've done some very standard dimensionality, reduction, and clustering that's performed in single cell. And so then this middle image, labeled Slide-seq reconstruction, what we've actually done is we've taken each bead on the array and we've colored it by the cell types identified in this dimensionality reduction. And so here you can see that the cell types reconstruct broad tissue architecture. For those of you who are familiar with hippocampal tissue architecture, you can see basically the CA1 neurons form a layer. You can see the dentate gyrus, and you can then see the oligo layer separating the hippocampus from the cortex. And for those of you who aren't familiar with the architecture of the hippocampus, you can see in this serial stained DAPI section that basically the labeled cell types and the structure of this tissue is recapitulated. The DAPI just kind of shows you where the cells are, and here we've assigned molecular identities to the cells. But we've done that in a way that requires no microscopy, right? And so the takeaway is that there's no microscope. We've turned the sequencer into a microscope. And instead of taking kind of like a one color or two color image, we're taking an image across all of the genes in the transcriptome at about 10 micron resolution. And so one of the first questions that comes up about Slide-seq is, what is actually the spatial resolution of this capture process, right? Where we take the tissue and capture the RNA onto the beads. And what we've done is we've come up with some assays to measure that directly by performing single molecule fish imaging in paired sections. So what we've done is we've taken two markers for the CA1 layer. That's the blue and green images here, SLIC17A7 and ATBP2B1. These are marker genes, and this is in situ hybridization. So we're showing you where the genes are being expressed in a serial section. And then we've actually performed Slide-seq on an adjacent, like just 10 microns away on an adjacent section, and we can go and measure kind of the width or the spatial extent of these features in single molecule fish, which is a direct microscopy measurement, and in Slide-seq, which is and for the same genes. And we find that the diffusion, or basically like how the width of these features are the same between Slide-seq and single molecule fish. And it's probably on the order, it's at least smaller than the bead size, which is about 10 microns. which suggests that we can improve the resolution of the technology by making smaller beads. And maybe just to examine the importance of spatial resolution, we performed Slide-seq on the mouse cerebellum. There's a question, we chose 10 microns because we had this intuition that 10 microns is about the size of a mammalian cell, and thus that might be the feature length scale that gene expression varies in tissue. But just a quick primer on the morphology of the cerebellum. It has kind of three primary layers. The Purkinje layer, cell layer, which is like the primary cell, the primary cell type of the cerebellum, here labeled P. The granule cell layer, labeled G here, and then the molecular layer, which is opposite the granule layer, side of the granule layer from the Purkinje cells. And here you can see at 10 microns, we performed Slide-seq and then assignment of cell types. You can see the reconstruction of the tissue morphology of the cerebellum. And you can also see accurate assignments of cell types and mapping to morphology. However, when we downsample the tissue to this data to 100 microns, so we just computationally aggregated beads, we can see that we can no longer assign cell types or recover tissue architecture. So we think that 10 microns is kind of a sweet spot where we can recover basically tissue features, and computationally we see that there's probably approximately 1 to 2 cells which cover every bead at 10 micron resolution. And so we think that Slide-seq, by collecting this high resolution transcriptome data, it allows a platform for spatial transcriptomics, and it allows us to reconstruct spatial architecture and connect it to molecular cell types, as I've shown you here. And so it allows us to localize the transcriptome with about one cell resolution in this untargeted way. And now I'll tell you quickly about 2 examples about how we can use it to discover new patterns of spatial variation within cell types, as well as how we can apply Slide-seq to visualize spatial gene expression changes in pathology. So first, I'll tell you about how we applied Slide-seq to discover spatial gene expression domains in Purkinje neurons of the cerebellum. And so I think because Slide-seq can do this kind of gene expression profiling in an unbiased way, we wanted to ask if we could systematically discover spatially varying expression patterns. And we wanted to do so in the cerebellum because when you sequence the Purkinje neurons, they're actually quite homogeneous in single-cell RNA-seq. But however, we've known for a long time that there's this thing, there's this histological banding of the Purkinje neurons when you sting them with this antigen called zebrin. So this is just like a fluorescent antibody stain against a surface marker, zebrin, and it labels some subset of the Purkinje neurons, and then there's this anti-correlated antigen PLC4, which labels a different subset. And it turns out that zebrin-positive neurons and zebrin-negative neurons have slightly different susceptibility to cerebellar disorders, as well as different electrical characteristics. But we don't recover this variation when we just look at it in single cell. And so what we did was we developed kind of a permutation testing algorithm to look at spatially varying gene expression across the transcriptome, but in a cell type specific way, just looking at Purkinje neurons. And when we applied it to the Purkinje neurons, we immediately found 2 clusters of genes corresponding to zebrin-positive and zebrin-negative neurons. And we can recapitulate the spatial these spatial populations in Slide-seq data. So here in this Slide-seq reconstruction, we've just plotted the expression of zebrin-positive genes and zebrin-negative genes only in the Purkinje layer. That's why you see this single line of beads or cells. And here you can see that that's validated in situ hybridization, the Allen. But beyond that, we additionally recover several previously unknown subdivisions of Purkinje neurons. And these divisions allow us to classify Purkinje neurons into at least 6 spatial subdomains. And these subdomain genes are enriched for channels and genes related to adhesion and connectivity, such as adherence. And also beyond just Purkinje's, we've discovered spatially and molecularly defined subpopulations of Bergman glia, granule cells, which were previously uncharacterized and could not be found with single cell sequencing alone. So we're really encouraged by this ability for Slide-seq to learn kind of like what are the determinants, what's the spatial variation in gene expression, and we're trying, and how does that relate to basically the variation that you see in single cell? And we're trying to currently follow this up in a larger study. So next, I think, as we motivated earlier in the talk, we really want to go and discover and visualize gene expression changes in pathology. And so, and I'll just talk to you about one example we did early on with Slide-seq, and we're actually trying to follow up with many other assays and examples or projects in our lab where we look at this. And so we use applied Slide-seq to a model of traumatic brain injury. And this is a kind of a cortical stab wound. This is a kind of a classic model, and it generates a robust inflammatory response known as astrogliosis. And what we wanted to do was we wanted to study the progression of cell type and gene expression changes in this process of glial scarring. So we applied this wound, and then 3 and 14 days later, we sampled the tissue with Slide-seq. So 3 days after injury, here, I'm showing you kind of the cortex of the mouse brain where we performed the injury. That's this image marked with the red arrow, and you can't actually see the injury at 3 days in just this histology image. But when we look at the transcriptome, so here we perform Slide-seq, we discovered kind of by looking at genes which are correlated, spatially upregulated at the site of injury, we discovered the mentin is highly correlated with the scar. And when we plot cell types, we can see basically at the site of injury, we see this enrichment in microglia, surrounded by signatures of mitosis. These are cells which are actively dividing in response to the injury. Surrounded, and this is just visualizing 3 cell types. There's, obviously neurons and other cell types which we're not showing here, but surrounded by astrocytes. And so 14 days after injury, here you can actually see this glial scar and this DAPI paired histology image. You can see that the red arrow marks the injury, and you can see this increased density in DAPI, which is many cells which have proliferated. Again, the mentin is highly correlated with this scar, and this is again in the cortex. And now when you look at the cell types, you can see that this architecture of the scar has developed, where in the very center you have these fibroblasts, which form this fibrotic scar, surrounded by microglia and astrocytes. And you can actually see that the neurons have been replaced. This is also a characteristic of this injury. The neurons are depleted within the scar. And putting it all together, we can actually reconstruct a spatial cell type profile across the wound, which kind of recapitulates the standard model of the glial scar, of this penumbra of astrocytes with fibroblasts in the very center. But additionally, we can also measure the infiltration of peripheral immune cells by measuring macrophage markers, and they show up in the very center of this scar. And beyond just cell types, we can look at the genes which are spatially upregulated at the site of injury. And here we've just plotted them as grouped by geoterms. These are kind of like metagenes. These are we've plotted like these genes which belong to these geoterms, which are upregulated at the site of injury. And we find that at 14 days, there's very little enrichment for mitosis at the scar, but I showed you I think there's this clear mitotic signature at 3 days and this rapid proliferation of cells. So I think you can see that the scar has reached steady state, at least in cellular proliferation between 3 and 14 days. But we see kind of terms for antigen presentation and gliogenesis upregulated as a function, spatially as a function of where the scar is. Very interestingly, I think when we look at immediate early genes, we found that they were, these are genes which are proxy for neuronal activity. And when we look at them just in neurons, we see an upregulation of IEG expression in a spatially localized region near the scar, about 1 millimeters on either side. And we think this is interesting because this suggests these changes suggest long-term changes in excitability in a spatially proximal way, which both is interesting from the biology of traumatic brain injury, but also from an experimental neuroscience perspective, because we often use stereotactic injection to gain genetic access to the brain, and we have to be careful then about these changes, peripheral changes, as a function of the injury that we cause. So I think, here I've shown you a couple ways that we've applied the data that we collected in Slide-seq. Since our initial publication, we've actually focused a lot on the production and most importantly, the yield of Slide-seq. Most importantly, we've significantly, I think one of the main barriers to adopting Slide-seq is actually the capture yield, which is how many unique molecules does each bead capture. And we've improved it just through a series of molecular biology optimizations, about almost 10-fold to approximately 1,000 unique molecules per bead. And this is similar, I think, to it's similar to the first generation single cell approaches, such as dropseq. And we think that there's no reason why we can't continue to push this yield. But I want to show some preliminary work showing how these improvements in the protocol can be applied to dissect out molecular and spatial trajectories in development. And so I think this new version of the protocol, which we put on bioRxiv, and all of the data has also been uploaded to bioRxiv. And so you guys can, I think, check it out. But first thing is that Slide-seq V2 allows for de novo clustering of the data. And here we've applied Slide-seq V2 on the E12.5 mouse embryo. This is an image of the mouse embryo with this white circle dotted line represents the region which we've sampled with this array, this 3 millimeter region. And here you can see almost 40 different cell types and states from this one section alone, recapitulating the known tissue architecture. But I want to focus on just the circle region, which is the developing eye. And just by unsupervised clustering, we can resolve 7 distinct cell types and states, which exist in this radial anatomical architecture, going in from the lens, cells of the lens, out to the edge, to cells of the cornea. And, we wanted to ask if, the eye is actively developing at this stage, and we wanted to ask if we can examine molecular trajectories of eye development. And so last year, I think Peter Karchenko's lab published a simple but, I think very useful concept of RNA velocity, which is in these dynamic processes, such as differentiation, we can infer the rate of change of gene expression by looking at the ratio of spliced to unspliced transcripts. And so when we apply this to the lens, we can generate these trajectory maps where the arrows represent the rate of gene expression change from one state to another state. And this is in UMAP space, which is just like a dimensionality reduction space. And you can see the lens plaque code has these strong flows to the lens, and the retinal epithelium has these strong flows to the more developed RGCs. And so, but more importantly, actually, we can perform these analyses directly on the spatial coordinates instead of UMAP coordinates. So this is a combination of spatial and molecular information. And now it lets us visualize the rate of gene expression change, but as a function of space. And so here we see similar patterns, but now we can recapitulate again this radial symmetry. And we see very strong transcriptional velocities in the developing retinal ganglion cells, and again, from the placode into the lens. And this is very preliminary work, but I think we're excited about developing algorithms to enrich for genes with very significant spatial changes. And then we see if we can connect that to the migration, spatial migration of cells or differentiation of cells. And so this is really, and we kind of developed we tried to apply these same analyses in a different context, the E14.5 embryonic cortex. Here, a state of the mouse brain, developing mouse brain, where many neurons are being actively born. And so here, just as a vignette, in this cortex, cells develop from the left to the right in this box region. The VZ, the ventricular zone, is where these neurons are born. And now we can actually, by examining kind of the spatially coherent paths of gene expression similarity, we can recapitulate both the direction and magnitude of this developmental process. And so what this allows us, and applying this, we can go in and discover genes that vary along this developmental manifold. And so we actually developed, we identified hundreds of genes that vary uniquely. We uniquely identified hundreds of genes which vary along this process. And then when we overlay these gene sets from human genetic studies of intellectual disability and autism, we find kind of an enrichment of intellectual disability genes earlier in development compared to autism, consistent with kind of previous microarray studies. And we also noticed that ID genes tend to be more highly expressed than the average spatially varying gene. And we think that these kind of data sets and analyses will be very useful in understanding the spatial-temporal localization of genetic risk variants in CNS tissues, especially as we go on and scale up the production of many developing cortex data sets with Slide-seq. And so I think in summary for this part of the talk, I've shown you how Slide-seq provides a platform for scalable spatial transcriptomics, and it allows us to reconstruct the organization of tissues and connect it to molecular cell types. We can also use it to discover new patterns of spatial variation within cell types. And it allows us to discover and visualize gene expression changes in both in pathology and in development, where there are very dynamic processes. Now, so far I've shown you results mostly from unsupervised clustering, where we take the same approach as a single cell. We cluster the beads by their transcriptional programs, and then we use marker genes to identify different cell types. But in many cases, we have actually great single-cell RNA-seq data sets and transcriptomic annotation. And what we'd like to do is be able to assign beads to the cell types on the array from these single-cell atlases. And this would allow us to ask questions about cell type organization or cell-cell interactions, and as I alluded earlier, spatial gene expression patterns, but also as a, I think excitingly, how does gene expression change as a function of your cellular neighborhood, or who your neighbors are, right? And, but I think one challenge that we face with all spatial transcriptomic data is that each bead is approximately the size of a single cell, but oftentimes there can be more than one cell per bead, maybe two, I think on average in Slide-seq data. And we can illustrate the effect of this with two cell types in the cerebellum, which are spatially colocalized. These are the Bergman glia, which is a supporting cell type, and Purkinje neurons. And so when we perform unsupervised clustering on them, we find clusters of beads which are predominantly glial markers or Purkinje markers. But we also kind of assign beads which have both Purkinje or glial character. And these beads basically are mixtures of these two cell types. And for cell type aware analyses, right, like how does gene expression change within a cell type, we don't want to say that these mixtures are one cell type or other. We actually really want to say there's some combination, and we want to know what the mixture rate is. And so we recently developed kind of a supervised learning approach to decompose these mixtures into single cell types. and from a single cell atlas. And this is really work done by Dylan Cable, and it's together with Rafael Irizarry. And I won't go into the details of the model since Rafa spoke about it in his talk, but you can check out the paper on bioRxiv. And you can check out the paper on bioRxiv. But the basic idea is, we want to estimate the contribution from individual cell types to each bead on the array. And we think that each bead is maybe a singlet or a doublet or a sparse a mixture of a sparse number of cell types. And so, for example, here in the Purkinje Bergman cells, there are beads which are singlets, just purely Purkinjes, and there are beads that are maybe a mixture of Purkinjes and Bergmans, and then there are maybe other cell types like granule cells. And we want to, what the model it does is it takes a single cell reference and then the Slide-seq data, and it outputs kind of for each bead, what combination of cell types and in what proportion best fit the observed data. And so we've performed kind of very detailed computational simulations with single cell and single nucleus data sets that demonstrates that we can classify cell types with this method. I won't go into the details here, but we can classify cell types very accurately. We can identify, if we simulate doublets, we can identify what are the cell types in the doublet and what proportion the cell types are and we can decompose them into the predicted proportion given a computational mixture. And so how does this important for Slide-seq data? Well, it allows us to use single cell RNA-seq references and project them onto Slide-seq data and assign and decompose cell types. So here again, we come back to the Bergman glia and Purkinje neurons, and we can show now that we can take these cells and we can classify them as singlets, where beads they're predominantly Bergman and Purkinje, but then also for the doublets, we can predict their doublet proportions. And these basically, when we do this follows very well with the ground truth of the marker genes. And so why is the ability to do this important? Well, because there's really been a lot of work that people have done in trying to detect spatially varying genes from spatial transcriptomic data. And usually these approaches are not cell type aware when they developed it. And thus, if you look at the topmost spatially varying gene from Slide-seq data, you might get a gene such as P2K2B. And it's clear from this histology image that it is non-randomly distributed in the hippocampus. However, when you examine the gene in the context of other cell types, you see that it's almost all the counts for this gene is in excitatory neurons. In other words, this cell type marker is a marker. This gene is a cell type marker for excitatory neurons. And obviously, excitatory neurons are non-randomly distributed in the cerebellum. And in fact, when you take genes in general to be identified as spatially varying, when you don't take into account cell types, they're almost all marker genes. And you can see that in this plot, the coefficient of variation across cell types for genes that are detected as spatially varying, ignoring cell types versus randomly selected genes. And while it isn't kind of important to discover cell type markers which are spatially varying, you can discover cell type markers in single cell data. What we really want to find out is how much of the variance in gene expression within a cell type is a function of the cell's spatial localization and as a function of tissue organization, right? And so there you need to go and discover gene expression variation within a cell type. And so using RCTD, we're beginning to be able to do that, right? So for example, here, once we condition on cell types, we can discover within cell type spatial variation. For example, here in CA3 neurons, I'm just showing you two example genes which define different subregions of CA3 that you can discover once you deconvolve the cell types from the spatial data. And we can also find changes in gene expression as a function of who your neighbors are. And this is something of a great focus for further algorithmic development in our lab. Here we did a simple test where we looked at astrocytic gene expression as a function of their neighbors. So astrocytes are kind of supporting cells in the brain. And when we look, we basically look, what are the genes that are differentially expressed when you're next to, say, a neuron versus, say, another astrocyte. And for example, here we find SLIC6A11, a GABA transporter, is expressed in astrocytes co-localized with excitatory neurons, which are these over here in this plot on the very right. Excitatory neurons are shown in blue, and astrocytes are shown in orange. And we know, sorry, the astrocytes which are next to excitatory neurons are shown in blue, and the astrocytes which are next to other cell types are shown in orange. And the bright spots are where the expression is, and so you can see it's highly enriched for the population in blue. And so we know this gene is associated with perisynaptic astrocytes, and this is just one example. In general, what we want to do is we want to condition on cell types to deconvolve the contribution of the cell type and cellular environment on cell state, right? That's kind of how you can understand systematically how do cell-cell interactions, or how does, where you are in the tissue influence your cell state. And so I think in this talk, I've shown you kind of a platform for understanding cellular circuits at the tissue level. And we've developed kind of experimental and computational tools for high resolution, high sensitivity spatial analyses of tissues. We've developed kind of molecular biology approaches where really increase the sensitivity of Slide-seq such that it approaches single cell technologies. And we've also developed ways to scale up this array production such that we can approach kind of tissue scale atlases, which I'll cover. And I think we've also shown you how we can leverage single cell atlases to assign Slide-seq data to cell types, which is really going to open up kind of these cell type aware spatial analyses to look at spatial differential expression as well as cell-cell interactions, things that we're really focused on in the lab. And I think now really we're prepared to generate complete atlases of mammalian tissues of cell type and gene expression in space. And we're starting with an atlas of the mouse brain and of the developing embryo. And I think, we should be able to generate a complete spatial transcriptome map of the mouse brain at a similar sampling of the Allen Brain Atlas at near single cell resolution. And we anticipate that takes something like 300 to 400 Slide-seq arrays. And so, having worked on basically experimental and computational aspects of this pipeline, I think we're very close and excited to getting to this next stage of actually now finally applying these technologies to realizing kind of the principles behind tissue organization. And I think, I think we're leveraging kind of these platforms for spatial genomics to be able to actually perform like genome scale measurements in real tissue contexts, right? Instead of having to dissociate and look at single cells. I think we're going to start to be able to make many measurements directly in the context of tissues. And, my lab and our lab is really focused on development and disease and understanding these processes. And we're actually developing kind of a lot of other tools which are complementary and paired with Slide-seq, including the ability to read out other modalities of readout and other like higher and higher resolution measurements of structure within tissues. And so thank you. I'm happy to take any questions. I want to really first thank the team who have worked on Slide-seq. This is a joint effort between Evan and me, and originally really led by two graduate students, Sam and Bob, and recently Evan Murray and Dylan Cable has really joined the team, and many others. Of course, I've just started my lab at the Broad just a couple years ago, and I'm also transitioning now to really, we're very interested in growing the lab. So if any of you guys are also interested, feel free to reach out. Thank you.
That was wonderful. Thank you.
Thank you. It was an interesting experience. I think
recording yourself.
Pre-recorded talk.
Right.
Yeah, I saw a lot of questions on the poll, but I thought I could answer them online, but it turns out it can't actually reply to I couldn't figure out how to respond to the questions directly, but I can answer them now. Sure.
Okay. Do you want to start at the top? Do you want me to? Shall I read it?
Yeah, sure. You can go ahead and yeah.
Okay. So the top most voted question is: have you compared the results from Slide-seq with those from the spatial transcriptomics technology offered by 10x Genomics? How similar or different are the data from the two different technologies?
Yeah, that's a great question. We've actually compared the data in our recent bioRxiv paper on Slide-seq V2, and I think the key distinction is that the 10x has spatial resolution about 50 microns. Each feature is about 50 microns, so about 25-fold by area worse spatial resolution. So obviously many more cells are captured per spot. By a per area capture, the data from Slide-seq has slightly higher sensitivity, but I think the 10x solution generates very high quality data as well. Now computationally, because you have a lot more cells per spot, it makes kind of deconvolving the cell types more challenging. And we've also, Rafa and I and Dylan have explored a little bit on the analysis on how to do that in our bioRxiv manuscript, and our separate bioRxiv manuscript.
So I noticed this question has been updated since I last asked it. There's a second question, as I guess like a part 2. Can this technology be extended to sequencing RNA from different parts of the neuron? What are your thoughts?
Yeah, yes, exactly. I mean, the beads then capture basically the RNA from, whatever part of the cell is on top of it. And obviously, if your synapses and dendrites and your neurons are in some stereotyped architecture, then it's easy for you to do that analysis. And we actually perform some of it to look for dendritically enriched RNAs. Now, because the resolution is 10 microns, you can't do that you can't say that you can detect the RNA from any given synapse, right? So it's kind of like at this resolution, you can enrich for these dendritic transcripts.
Thank you. So I'll move on to the next most highly voted question. What types of computational algorithms and or software do you recommend for clustering, say, for spatial beads?
Yeah, that's a great question. So, we almost, you can take just a lot of data here was done with just like standard Seurat clustering for unsupervised clustering or using NMF decomposition. We also, have recently developed this new algorithm for aligning single-cell RNA-seq data sets to Slide-seq data, which we think, if you have the single-cell RNA-seq data, is definitely the way to go because it's just so much more powerful.
Great. Next is, if you are studying a tissue with no prior information about spatial gene expression patterns, how would you check if an observed pattern is an artifact, a secondary biological effect, or the biological effect of main interest? The question is by The question is by Leo.
Yeah, that's an interesting question. Now, we've performed quite a lot of ground truthing of Slide-seq against fish to basically show that Slide-seq data just recapitulates, and I think Visium also recapitulates, the spatial expression pattern for Slide-seq at 10 microns. Now, how do you, test to see if differential expression at that resolution is meaningful to your biological system? I think the way to think about it is that there's two ways to think about it. One is that I think Slide-seq is a great way to generate, like, do hypothesis generation, just like single cell is a great way to generate hypothesis generation. And if you do see kind of differential expression spatially or in single cell differential expression between two clusters, you obviously still have to follow that up with either a functional experiment or other lines of evidence. So, you know, to get at the biology, which is obviously important, I think, you know, you need other ways to attack the problem.
Makes sense. So the next question is, how is a spot within the tissue mapped onto a spot on the bead slide? Is the tissue fixed onto the slide? Is there a danger that the beads might become overloaded with tissue nucleic acid?
Yeah, that's a great question. So, you know, it's an interesting process. The beads are kind of on the surface, and then we take this tissue and we section it directly onto the tissue, onto the beads. Right, and there you can take a picture of the tissue on top of the bead. So you can kind of have a little bit of, you can have that registration. Most of the time we don't do that because the Slide-seq data, the transcriptome data recapitulates the tissue architecture anyways. But for example, if we want to do reconstruction of many serial slices, we kind of take these registration images. Now, whether the beads become overloaded with nucleic acid, I don't think that's the case where we are now. I think we're more worried that the beads are, you know, there's a lot more nucleic acids to capture, and we're in the process of like figuring out how to actually capture those nucleic acids from the tissue. We think the beads only capture a small fraction of the nucleic acids from the cell, something like 5 to 10%.
Interesting. Okay, well, I believe we are at time. Give Fei another round of applause. Thank you so much.
Thank you. Thank you for hosting me too.
And we wish everybody a great continuation of bioconductor today.